So today's focus is about genome editing. So that's a disruptive and groundbreaking technology used to edit DNA in living cells. Okay? So some new tools of the genome editing has already changed the landscape of science and the way scientists do research. So in my first talk, I'll give you some overviews and background information of genome editing and also the basic science behind. And the other two speakers will introduce you the available tools, potential applications, ethics concerns, and the future directions. So what's genome editing or gene editing and how it works? How does it work? So let's start by talking about genes and DNA. So the cell is a basic subunit of the human body, and within each cell is a compartment called nucleus, which contains the entire genome. And the genome is composed of the chemical DNA, which will be transcribed into the RNA molecules, then translated into protein. Okay? So the DNA is an instruction manual for building every human cell. So that's a code of life. The precise sequence of DNA, that's 3 billion individual nucleotides in human, carries the information of the genome, which encodes about 20,000 different genes. So in human, these genes can influence many of your characteristics, such as your height and the color of your eyes. So as Stephanie mentioned, the humanity has a long history of figuring out ways to modify genes. So we have been crossing breeding the plants and animals to make them better to eat for thousands of years. So now with the genome editing, we have made a great leap forward because this new technology is faster to use and relatively simple and very cheap. Okay? So its applications are incredible, already transforming the genetic research and has great potential to make big changes to our lives. So in my talk, I will first introduce you the basic scientific background of genome editing and also the history of its discovery. So as we know, the DNA is a repository of the genetic information in each living cell. However, so its integrity and stability is essential to life. However, the DNA damage occurs in our body very frequently. Okay? In human cells, both the normal metabolic activities inside a cell and the environmental factors such as radiation can cause the DNA damage, resulting as many as 10,000 to 1 million individual molecular lesions per cell per day. So while this constitutes only a tiny fraction of the entire genome of 3 billion individual nucleotides, the unrepaired lesions in critical genes such as tumor suppressor gene can impair cells' function and increase the likelihood of tumor formation, for example. However, the cells develop complex mechanisms and a variety of strategies to fix them, to identify and solve the DNA damage and maintain the genome integrity. So the DNA repair machinery is constantly active and efficiently responds to the DNA damage. For example, so the double-strand break is one type of DNA damage. So the 2 DNA strands are broken. So the double-strand break can be repaired in different ways. It can be repaired through either the non-homologous end joining or the homology-directed repair mechanisms. Okay? So the non-homologous end joining can induce random mutations in the genome. Could be the insertion, could be the deletion. We call them indel mutation. So the DNA double-strand break can be repaired through the non-homologous end joining, but the result is the random mutation will be induced. Okay? So these mutations may disrupt gene function. Okay? So, but the homology-directed repair with the DNA donor template through the homologous recombination can lead to the precise repair. So actually, the cells use the sister chromosome to recover the genetic information through the homologous recombination. Okay? If the DNA donor template contains some alteration, nucleotide alteration or insertion, so then this alteration insertion can be recombined into the genome through the homologous recombination. So the scientists take advantage of these types of natural DNA double-strand repair mechanism to achieve genome editing, to change the DNA through either the random mutation or the homologous recombination. Okay? So what do you need to do? If you want to disrupt a gene, you can induce a non-homologous end joining. You will induce a random mutation, and this mutation may disrupt gene function. If you precisely modify a gene, you can design and supply a suitable DNA donor template that carries the mutation that you want to introduce into the genome. Okay? So that's the basic science behind the genome editing I want to introduce earlier. And the scientists make use of these kind of mechanisms, the natural DNA double-strand break repair mechanisms, to achieve genome editing. So in old days, if you want to modify a gene to introduce or knock out the fragment or introduce a mutation, we rely on the spontaneous homologous recombination occurred in cells. Okay. So to achieve this, we need to first making a gene targeting vector containing the homologous arm that match the sequence of target gene. So then the recombination, the homologous recombination can occur between the homologous arm and the target gene. So the sequence need to be seen in the homologous arm and the target gene. And you also need a selection marker in the targeting vector. Then you can select out the cells that undergo homologous recombination. However, the efficiency is very low because the spontaneous homologous recombination is a very rare event in normal cells. So at time of 15 years ago, if you want to modify a gene, it's a very big task. You have to make a complex gene targeting vector, contain the homologous arm and the selection marker. Then you need to select out the cells that happen to undergo the homologous recombination. So the efficiency is very low and is very, very time consuming. So the way to enhance the homologous recombination is the key to efficiently modifying the genome. So as I mentioned earlier, the DNA double-strand break can induce the homologous recombination, right? So then people try different ways to induce a double-strand break in order to induce a homologous recombination. But you have to induce a double-strand break at a specific locus precisely. Otherwise, if you induce double-strand break everywhere, the cell will die. So the one strategy people use is to pre-insert a real restriction enzyme site sequence into the target locus. So this specific sequence was inserted into the target locus that you want to cut. So then you can use a nuclease, it's an enzyme that recognizes this sequence, then you can cut the DNA, make the double-strand break. And this strategy works very well, can enhance the efficiency of homologous recombination. So after you induce a double-strand break by this enzyme. However, there is a problem. Because you will take more time and effort to pre-insert this sequence into the genome. That's not easy. So that's why very few like this strategy, and people develop a better one. So people engineer the nucleus, the enzyme. So they engineer the nucleus to make it composed of two domains. One is the DNA binding domain that can recognize and bind to the specific sequence of DNA. The other is the DNA cutting domain, so that's the nucleus actually, that can cut the DNA, make the double-strand break. So then the chimeric nucleus enables precise and efficient genetic modification by inducing the targeted double-strand break, okay, that stimulates a homologous recombination. So basically, it fills the DNA binding domain and the DNA cutting domain. So the DNA binding domain will bring the DNA cutting domain to the specific locus to cut the DNA, make the double-strand break, induce homologous recombination. So that's a strategy people use to enhance the efficiency of homologous recombination. So the first engineered the genome editing nucleus is called the zinc finger nucleus, as Stephanie mentioned. The zinc fingers are the most common DNA binding domains found in eukaryotes. So they typically comprise of multiple modules that interact with different nucleotide triplets. For example, you can see here, one module of the DNA binding domain of zinc fingers can recognize and bind to the specific sequence of TTC. So then the next module of the DNA binding domain can recognize and bind to the sequence of TAC, a different sequence. So then you can create artificial zinc fingers that recognize any specific sequence of DNA by simply assembling different modules to form a new artificial DNA binding domain. So this artificial DNA binding domain is fused to a nucleus called fork1, so that can cut the DNA, make the double-strand break. So because fork1 functions as a dimers, a pair of DNA binding domain is required. One recognizes the sequence upstream, one recognizes the sequence downstream of the site to be targeted. Okay? So that's how zinc fingers work. So the DNA binding domain bring to the nucleus to the specific locus to cut the DNA, make the double-strand break. And later on, a better one was developed. called TALEN, which is similar to zinc finger. It also uses a DNA binding domain to guide the focal nucleus to cut the DNA, to cleave the genome at a specific locus. But instead of recognizing 3 nucleotides, each module in the TALEN recognizes single nucleotide, not a trinucleotide as zinc finger does. So designing the TALEN is more straightforward than the zinc fingers because you only need 4 different modules to assemble, okay, the ATCG. After many years research and development, the zinc fingers become the most popular genome editing tool. But after 2010, more and more people start to use TALEN. Both the zinc fingers and TALEN can enhance the targeting efficiency by inducing the precise double-strand break and the subsequent homologous recombination. But both of them require assembly of pair of proteins that recognize the sequences flanking the target site. And this step is very time-consuming. So that's why, even with the zinc fingers and TALEN, genome editing in a research lab is still a big task until the appearance of new genome editing tool known as CRISPR, so which is so powerful and easy to use. So unlike the zinc finger TALEN, which rely on the protein and the DNA interaction, the sequence specificity in CRISPR is guided by RNA. It relies on the RNA and the DNA interaction. Okay, so the major breakthrough was made in 2012 when people found the Cas protein in the CRISPR system is actually an RNA-guided nucleus that can be designed to recognize and cut the DNA in the test tube. So soon afterwards, after the CRISPR was discovered, and the CRISPR was causing a major revolution in biomedical research. So unlike other genome editing tools, the CRISPR is so easy and very fast to use and very simple and very cheap. And it has swept through the research labs around the world within very short time. So some scientists even comment CRISPR has completely changed the game of biology. So after scientists show that CRISPR can be used to edit the genome, the use of CRISPR spreads quickly. You can see the sharp rise of the CRISPR publications, research publications, the patents, and funding. Okay? So billions of money from both government and industry has been pouring into the CRISPR research. Okay. So now let's see why CRISPR is so powerful and what is CRISPR first, and how CRISPR works and why it's so powerful. I think you know bacteria, viruses can cause human diseases, and we know how the bacteria can fight off how human can fight off the bacteria with antibiotics, and how the virus, how we can prevent the infection by both bacteria and viruses using the vaccines. But actually, the bacteria and the viruses have also been fighting each other for very long time. So the virus for bacterium is also called bacteriophage or phage. Okay? So this type of virus, the bacteriophage, so lands on and attach to outside of the bacterium. So then it will inject its viral DNA into the bacterium. Okay? So if the bacterium doesn't realize the viral DNA is not its own, it will follow the viral DNA, the information in the viral DNA, to make more viruses. So then these viruses will destroy the bacterium, then go outside to infect more. So basically, the virus hijacks the bacterium to make the bacterium to make more viruses. Okay. So most bacteria that get infected by virus will die. But if the mutation happened in the bacterium DNA, okay, so it can fight off some few lucky bacteria can fight off the virus and survive. So it's very interesting studying the way the bacteria and the viruses fight to each other has led to the discovery of the exciting new genome editing tool, CRISPR. So let's see what happened in the history. The history of CRISPR began long before we know it's so powerful. So in 1987, some scientists studied a gene in bacterium and identified a curious set of 29 nucleotide repeats downstream of that gene. Okay? And unlike the most repetitive sequence in the genome, that's typically tandem repeats, these 29 nucleotide repeats were interspaced by some spacer DNA. So let's see here. You can see a sequence here, and this sequence is followed by a nearly same sequence in reverse. We call it a palindromic sequence. This palindromic sequence was named as direct repeats later on. And after this palindromic sequence, it's a sequence of random non-repetitive sequence. We call it spacer DNA. Okay? So that is a repeat of the same palindromic sequence followed by a different spacer sequence. So then another repeat of the palindromic sequence followed by another different spacer sequence. So that's a very interesting pattern of repeats in the genome, never reported before. Okay? So the direct repeats are interspaced by some random sequence. We call them spacer. So why this pattern is very interesting, but very few scientists took much notice about this finding. Then over the next 10 years, the people found a similar puzzling pattern. The additional repeat elements are reported from genomes of bacterial strains and archaea strains. So this type of repeats actually present more than 40% of sequenced bacteria and more than 40% of sequenced archaea strains. But most of scientists assume these sequences, all the sequences, actually are junk DNA. They consider this DNA are junk DNA. So in 2002, it was named as CRISPR, standing for clustered regularly interspaced short palindromic repeats. That's how CRISPR got its name. So the CRISPR array contains many repetitive sequence, direct repeats. These direct repeats are interspaced by the spacer DNA. The spacer DNA are random, non-repetitive sequence. Okay? People also found a class of genes are conserved and adjacent to the CRISPR array. They named these genes as CRISPR-associated genes or Cas genes. Okay? But nobody knows their functions, what Cas gene or the CRISPR array is doing. The breakthrough was made in 2007 by a food company called Danisco. Okay? So the virus infection is a potentially big problem in dairy industry. And because their production depends on the bacteria to produce yogurt and cheese. Okay? But the virus, the bacteriophage, okay, can destroy their precious bacteria strains. So for this reason, a number of employees of this company are engaged in research. When these scientists compare the sequence of the non-virus resistant bacteria and the virus resistant bacteria. So the non-virus bacteria, resistant bacteria means they are not resistant to the virus. But the virus resistant bacteria, so they can fight off the virus. So when they compare the sequence of CRISPR regions, these non-virus resistant bacteria and the virus resistant bacteria, they found the only difference between them was the virus resistant bacterium had an extra spacer sequence in the CRISPR region. So the only difference between them is here, the extra spacer sequence. And very interesting, the extra spacer sequence exactly matches the sequence of the viral DNA. So part of the viral DNA has the same sequence of the extra spacer in the CRISPR region of the virus-resistant bacteria. Right? So if they delete the spacer sequence from the virus-resistant bacterium, the bacterium lost its resistance to the matching virus. But if they add the spacer into the non-virus resistant bacterium, so the bacterium would be resistant to the matching virus. So then they conclude the spacer sequence is the key, provides the resistance to the virus by saving some of the viral DNA during the infection. Okay? So this will allow them to remember the virus. So these are very critical experiments, and this critical experiment has figured out how CRISPR works to prevent the virus from destroying the bacterium. Okay, so let's see. During the then many other following studies after this finding revealed the detailed mechanisms of CRISPR. So you can see during the infection of the virus, so the virus will attach to the bacterium, inject the viral DNA into the bacterium. So the bacterium will clip a bit of the viral DNA and insert it into its CRISPR region of its own DNA. So then a new spacer will be created. Okay? So a new space will be created after the first round of infection. Okay? So if the virus comes back, so the bacterium will make the RNA from the CRISPR region specific for the virus. So this RNA will pair up with the Cas protein and guide the Cas protein to the invading virus. And the Cas protein is actually a nucleus that can cut the DNA, make the double-strand break, so the Cas protein can destroy the viral DNA. Okay? So no more viral DNA, no more viruses. So basically, people understand now the CRISPR is actually a bacterial adaptive immune system. The CRISPR array serves as a immune memory and the defense mechanisms, and the spacer sequence facilitates the defense against the virus infection by the RNA and the DNA matching. So the RNA transcribed from the spacer sequence can match the viral DNA by the RNA-DNA binding, so then guide the Cas protein, the nucleus, to cut the viral DNA and destroy the invading bacteria. So that's how the CRISPR system works in bacteria. So from 1987 to 2011, it takes us nearly 25 years to understand the CRISPR is a bacterial immune system and how it works. But after that, the CRISPR research has exploded. A milestone paper was published in 2012 by Jennifer Doudna at UC Berkeley. She actually visited Hong Kong in 2017 and received the honorary doctorate degree from Hong Kong U. So in her milestone paper, she showed that she could control where the Cas protein went to do its job to cut the DNA. She basically mixed the spacer sequence and the Cas protein in a test tube and shows this complex can target the DNA and cut the DNA in the test tube, completely in vitro, without bacteria. It's not happening in the bacteria, just in the test tube. So let's see what she did. So first, let's see again what happened in the bacterium. So the CRISPR array will be transcribed into an RNA molecule, the pre-cRNA, containing multiple repeats and many spacer DNA, random spacer DNA, and each spacer DNA can match the DNA of the virus. Okay? So this pre-cRNA will be processed into a mature version, and each mature version contains only one spacer DNA and one repeat. We call it a guide RNA. So then this guide RNA will form a complex with Cas protein, and will guide the Cas protein to the targeting viral DNA and make a double-strand break and destroy the bacterium. So that's what happened in the bacterium. So what Jennifer did is she mixed the artificially synthesized Cas protein and the guide RNA into a test tube. It's not bacteria, just into a test tube that contains the targeted DNA molecules. So then the Cas protein and the guide RNA will form a complex called Cas RNA complex. So this complex will find and bind to the specific sequence of the DNA molecules by the RNA and the DNA binding. So then as a result, because the Cas protein is a nucleus that can recognize the target sequence DNA and cut the DNA, make the double-strand break. So now you can imagine how easy to target and cut DNA molecules. You only need a Cas protein and a guide RNA. So what do you need to do? Just design a guide RNA with a specific sequence that match the sequence of the target DNA molecules, right? So that's a very critical experiment. Everything can be done in a test tube, not in the bacteria. So soon afterwards, another important person of CRISPR appeared. In 2013, Fen Zhang from MIT quickly applied this technique to edit DNA in human cells. So show that the CRISPR has high efficiency to bind and target the DNA in human cells. So that's first demonstrating that the CRISPR was initially found in bacteria, could be used to engineer the DNA in cultured human cells. So in his experiment, he didn't do the experiment in the test tube. He did in human cells. He expressed the Cas gene and the guide RNA in the cultured human cells to make them to cut the DNA, make the double-strand break. Okay? If you recall back what I showed you at the beginning, the double-strand break can be repaired, right? Can be repaired through either the non-homologous end join or the homologous directed repair. If the non-homologous end join happen, so the random mutation will be induced. Could be insertion, could be deletion. and this random mutation can disrupt gene's function. But if you supply a DNA donor template, then the homology-directed repair mechanisms will be induced. And if your donor template carries an alteration or insertion or mutation, this change can be incorporated into genome through the homologous recombination. So now you can imagine that how easy to edit a gene. If you want to just disrupt a gene, you just make a double-strand break at a specific locus. Then through the non-homologous end join, the gene will be disrupted by the random mutation. But if you want to modify a gene precisely, you need to design a DNA donor template that carries a mutation or alterations that you want to introduce into the genome. Okay? So by these strategies, you can easily modify a gene to what you want. So that's why after Fenjiang's paper was published, so the use of the application of the CRISPR in cultural cells, in human cells or other type of mammalian cells quickly spreads, swept through all the research labs around the world. Nearly every scientist start to use that. Okay, just a few months after this publication, the scientists already applied this technique to make transgenic mouse model. Okay? And this revolutionized the mouse genetics. The mouse is a major animal model used for biomedical research. So in old days, if you want to modify a gene in the mouse to make a mouse model, you first need to make a complex gene targeting vector, as I mentioned, carrying the homologous arm, the selection marker. Then you need to electroporate this targeting vector into the embryonic stem cells. You have to screen the embryonic stem cells to select out the cells that happen to undergo homologous recombination. So efficiency is very low. But after that, you can inject the embryonic stem cells into the blastocyst of the mouse, that's early embryo of the mouse, to produce a transgenic mouse model. So that's in old days. But now you can skip the targeting vector and embryonic stem cell procedures. You can simply inject the Cas protein, guide RNA, and the DNA donor template into the mouse zygotes, the one-cell embryo, the fertilized egg. So then you can produce a transgenic mouse model. So time is largely saved, and the efficiency is greatly improved. So when I was a student, it's a big deal to make a transgenic mouse model. I made my first annual model. Cost me nearly more than a year for my PhD to make the first transgenic mouse model. But nowadays, only a couple of months. And actually, most of the time, you are waiting for the mouse to grow up. Okay. So after mouse, the CRISPR has been shown to have a high efficiency in many species, from plant, invertebrate to vertebrate, in fly, in fish, in rat, in cow, in primate, nearly every species. It's highly useful for both scientific and economical purposes, because particularly for some species, because it was impossible to edit their genome in old days, because we don't know how to culture their embryonic stem cells. Okay? So, but now you can just edit their genome in their embryos, so the thing become very easy. So that's why CRISPR is so powerful, completely changed the game of biology. Okay, so over the course of discovering and using CRISPR, there are many great pioneers and unsung heroes. So they continue to push the frontiers of the CRISPR research, or they are no longer alone. So many of the thousands of scientists around the world now engaged in CRISPR research to continue to further elucidate its biology, improve the efficiency, and apply it to a broad range of biomedical problems. And recently, a scientist from Harvard even developed a new genome editing tool called base editing. And I believe that our next speaker, Ellen Wang, will talk about this new tool of genome editing. So it's very, very impressive. Many of the heroes of CRISPR did their seminal work at a very young age. And some of them are even before age of 30. So probably because young people are more willing to take risks, I guess. And actually, many biomedical breakthroughs often come from the completely unpredictable origins. So some early pioneers of CRISPR were not on a quest to edit the human genome. So they are not aiming to edit the human genome. So their motivations were a mix of personal curiosity and probably the industry applications. For example, they want to understand the bizarre repetitive sequence. They want to improve the yogurt production. Okay? But this type of motivations with courage will eventually drive you to succeed. Okay? So I probably will stop here. And thank you very much for your attention and this opportunity to share the story of genome editing. And I think our next speakers will give you more about this amazing tool. Thank you very much.